All right, welcome back. Today we're going to start looking at um, genetics, and we're going to start off with basic Mendelian genetics. We'll start off concept 14.1. So Mendel used the scientific approach to identify two laws of inheritance. So um, this is all exploring what principles account for the passing of traits from parents to offspring. So there were two general ideas competing at the time. So one is this idea of blending, that the genetic material from the parents somehow blend together, kind of like paints. So you would have blue and yellow paint blend together to create green. The competing idea was this particulate hypothesis that parents pass on discrete inheritable units. And this is ultimately where we get the idea of genes from. So, person who kind of launched this or answered this major question is Gregor Mendel. He was a monk back in the 1800s. He essentially became a monk because he wanted an education because he was the son of a farmer, right? So if you wanted an education at that point in time and didn't have money, the church was a good way of going. So here he is along with his fellow monks, and you can see he is staring at a sprig of fuchsia. So even in this picture, he's demonstrating what he did. All right, so to start off with, Mendel's discovered the basic approach of heredity by breeding garden peas in carefully planned experiments. So why peas? Well, peas were available in many different varieties. They have a short generation time, so he could breed plants, grow them, and then see the results fairly quickly. Also, it produces large numbers of offspring. This allows for better statistics. And mating could also be controlled, so you could decide whether you would allow a plant to self-pollinate or to cross-pollinate. And I'll come back to this idea. Now, a couple of definitions here. A heritable feature that varies among individuals is called a character. So an example of character would be the color of a flower, right? Another thing to think about is each variant for this character can be called a trait. So purple is one trait, white is another trait. So pea plants could have purple flowers or white flowers. That idea of flower color is called a character. The actual real color is called the trait. Now, the reason why he was able to utilize this plant is the flower of a pea plant has two parts. One is the stamens, which release pollen. Pollen is equivalent to sperm. The carpel holds the egg, which needs to be fertilized by the pollen. So consequently, if you went in to the parent plant and before the flower had fully developed, you cut off all these stamens, you would not release the pollen. Consequently, you could take a paintbrush, go to a plant that did have stamens, pick up the pollen and transfer it directly, and in this way you can control breeding. He also utilized varieties that were called true breeding. So he had varieties of pea plants that always had the same characteristic. They always had purple flowers. They always had green colored peas, or they were always tall, or they were always short. All these individual characteristics would always be true for this plant. And this is achieved by essentially inbreeding that plant until all the variety goes away. And this is very common in breeding different types of plants. All right, so a typical experiment is something that involves taking two true breeding varieties and doing a cross-pollination procedure called hybridization. So he would create a hybrid of two true breeding varieties. Those true breeding varieties are referred to as the P generation or the parental generation. The hybrid offspring of that P generation is called the F1 generation. And you would see particular characteristics associated with this F1 generation. If you take that F1 generation and allow it to self-pollinate or to cross-pollinate it with some other type of F1 hybrid, you'd create an F2 generation. And sometimes you can see some characteristics that only show up in the F2 generation. So, one of the laws we have is the law of segregation. So when Mendel was contrasting true breeding white and purple flower pea plants, all of the F1 generation were purple. So you would take white and you would take purple, you would crossbreed them, and they would always be purple in this F1 generation. That is telling you something about the genetic principles. It tells you that purple is what we call dominant. But there's something funny that happened when he went to the F2 generation. If you allowed those F1 hybrids to self-pollinate and looked at the F2 generation, you'd recognize that there were some purple flowers, but a very small percentage had white. So now, with two generations past, you could all of a sudden see one of the characteristics or the particular trait of this white flower coming back. He also discovered that there is a specific ratio that occurs in this F2 generation, specifically a 3-to-1 ratio. So, looking at this graphically, we have two pea plants. One has purple flowers, one has white flowers. You trim the stamens off of one so it can't self-pollinate. You then cross-pollinate with a paintbrush. This is the pea generation. Its offspring has all purple flowers, right? No matter how many times you did this, you always get purple flowers. You do it again, allow these F1 generation or these hybrids to self-pollinate, you now get something different. The F2 generation has mostly purple flowers, but also does have a few white flowers. So something is coming back. And it specifically happens in a 3-to-1 ratio. So here we have 705 to 224. So he did this with several different characters with multiple traits and always found the same thing. So here we have purple flowers versus white flowers. Here we have yellow seeds versus green seeds, round seeds versus wrinkled, and all these different characteristics. And if you notice, he did this a lot. So here is had some 6,000 offspring versus 2,000 offspring. So he didn't skimp on doing this just one or two times and calling it quits. He did this extensively. But every time you do it, you always end up with the exact same ratio. Notice it's never perfectly 3-to-1, but it's always clustered around that 3-to-1 ratio. So Mendel developed a hypothesis to explain this 3-to-1 inheritance pattern that he saw in the F2 generation. So four related concepts makes up this model, and these concepts can be related to what we now know about genes and chromosomes and DNA, because remember, he had no concept of DNA at the time. So the first thing is alternative versions of genes account for variations in the inherited characteristics. So we'll call these alleles. So there is an allele that accounts for a particular version of a character or a particular trait. So second, for each character, an organism inherits two alleles, one from each parent. They might be the same or they might be different. So one allele might be white, the other might be purple, they may both be purple, but you only get one from each parent. Third, if two alleles at a locus differ, then there will be one dominant allele, which will determine what that organism looks like or its appearance. The other, what we call recessive allele, will have no impact on appearance. And we'll explain why that is here in a moment. Fourth, we have the law of segregation. So the two alleles for an inheritable character separate during gamete formation and end up in different gametes. So you may have an allele for a white flower, an allele for a purple flower, but when you produce gametes, each gamete will only have one or the other. So moving this into a genetic standpoint, here is a piece of DNA. It has a gene on it. That gene produces an enzyme, and that enzyme is responsible for synthesizing the purple pigment. The white flower version of this has the same gene at the same location on a chromosome, but it may have a different genetic sequence. So here we have CTA, here we have ATA. This produces a protein that is a non-functional enzyme. So consequently, if you only have this version, you cannot make the purple pigment. If you have one of both, you will still have a purple flower because you still make enzyme that will produce that purple pigment. So in this case, just having a single allele is enough to result in a purple flower. So coming back to this idea of purple and white flowers, this purple flower has a genetic makeup of capital P and capital P. It's a true breeding, so it has two alleles that have functional enzymes. The white flower has little p, meaning that it has two copies of the defective enzyme. We cross those together, and all of these flowers are going to have one big P from one parent and one little p from the other parent, because that is the only possibility. It will be purple because it still has a functional enzyme, and that one copy of that functional enzyme is enough. If you then cross this F1 generation together, where it is always big B, little p, you have four possible combinations that can come out of that. So recognizing that when you make gametes, you can only make a gamete that has a big P or a little p. You could have the father giving you a big P and the mother giving you a big P. You could have the father giving you a big P and the mom giving you a little p. Consequently, you could have the father giving you little p and the mom giving you big P or little p. In this case, 3 out of the 4 of these are purple because they have one copy of the functional gene. One copy here, or 1 out of these 4, will actually be white because it contains both little p's or both non-functional versions of the enzymes. We will pick this up again in part 2.